Hello, this lesson is on meiosis. So we just got finished studying mitosis. Mitosis, of course, is how cells divide and how you became one fertilized egg cell to 300 trillion cells that you have now. But all of your cells have the same number of chromosomes as the original. So then how do we make more humans on the planet, or how do plants make more plants? Well, there are two cells in your body that have to undergo not mitosis, but meiosis. And those two cells in your body are egg cells, if you're female, and sperm cells, if you're male. Okay, so you should have gotten a Cornell note sheet that says meiosis, making egg and sperm cells at the top. There is notes on the front side, and then the drawing is on the back side. Okay, we're first going to read a little bit out of the textbook, and then you're going to take notes in this section based on what we read in the textbook. And then we'll fill in the pictures on the back, and then you'll do questions at the end on the left side. So first, open your book to the meiosis section in your book. It's chapter 6, section 3. Go ahead and pause right now so that you can go and get your textbook, and then you can read along with me. So chapter 6, section 3 is titled Meiosis. The key concept is that meiosis and sexual reproduction allow for the combination of genetic material from two different cells. Offspring receive genetic information, or DNA, from their parents. What are offspring? Right here. What are offspring? Babies. That's all. So offspring receive genetic information, or DNA, from their parents. In sexual reproduction, or in asexual reproduction, one parent contributes genetic information to its offspring. The offspring has the same genotype as the parent. In sexual reproduction, two parents contribute genetic information to their offspring. I'm going to stop there. Your job right now is to write one sentence on your notes that summarizes that information right there. So in here, in this section, you're going to write a sentence. Oops, sorry about that. In this section that summarizes what you learned in that section. So I might write a little bullet right here, and then okay, obviously you don't write that, you write the actual sentence. So pause it right now so that you can write your sentence. Alright, so the next section says, so I'm starting right here at the word before. Before sexual reproduction can occur, each parent must reduce his or her genetic material by half in a process called meiosis. Therefore, when the genetic information from two parents combine, the offspring will have the same amount of genetic information as each of its parents. Okay, this is now your second sentence that goes on your notes on the first page. So back here, you might skip a line, and then your second sentence goes here. Kind of capture the main idea of those sentences in the book. Don't write some just random sentence from that section or that highlighted area. Write it in your own words. Okay, pause it so you can write your section or write your sentence. All right, going on. I'm going to start right here. So genetic information is located on structures called chromosomes. The next section talks about chromosome numbers. Most species have a specific number of chromosomes in their body cells. For example, human cells usually have 46 chromosomes, corn cells usually have 20 chromosomes, and dog cells usually have 78 chromosomes. Most of the time, the chromosomes are spread out in long strands. Before a cell divides, the chromosomes in the cell condense and become shorter and thicker. Now this is your next section. Try to capture the one sentence that captures the main idea from these sentences in the text. Pause it right now so you can write your sentence. All right, going on. The next sentence in here, I'm starting right here. Figure 1 shows the 46 chromosomes of a human body cell that are visible during cell division. Your extra credit on this Educreations is to write what figure 1 is showing in the top right corner of your Cornell Notes paper. Alright, so you should have now 3 sentences worth of information, okay? Now we're going to flip to a couple of pages down in this section. We're going to flip to the second to last page in this section and talk about the meiosis in general. So flip your book to this section, or this page, and now we're going to start right here reading. Sex cells are made during meiosis. Meiosis is a copying process that produces cells that have half the usual number of chromosomes. Each sex cell receives 1/2 of each homologous pair. So now go back on your notes and write now your 4th sentence on your Cornell Notes. Pause it right here. Okay, continuing on. I'm going to start right here. For example, a human egg cell has 23 chromosomes, and a sperm cell has 23 chromosomes. The new cell that forms when the egg and the sperm cell join has 46 chromosomes. So now write one sentence that summarizes these sentences. Go ahead and pause it. Alright, going on. Now we're down here at the steps of mitosis. Steps of meiosis. The steps of meiosis for a cell that has 4 chromosomes is shown in figure 4. Before meiosis begins, each chromosome is copied. The original chromosome and the new form chromatids, which are joined together. The chromosomes shorten and thicken, which makes them visible under a microscope. During meiosis, the nucleus divides twice. The first time the nucleus divides, the homologous chromosomes separate. The second time the nucleus divides, the identical chromatids separate. Finally, meiosis results in 4 sex cells, which have half the number of chromosomes as the original cell. I know, that's a lot. And am I going to make you write one sentence for that whole thing? No. I want you to focus on this one right here. So pause it, go back, and write a sentence in your own words that summarizes that main topic, that main sentence right there. Alright, so now you have bullet items here on this page. Now flip over to the back, and now we're going to start coloring. So you will need just 4 colors and a regular pen or pencil for this. So you pause it right here and go and get 4 colors and then start it again when you're ready. So this first box is your interphase. You know this one, okay? But we're going to draw the interphase after the chromosomes have already formed, so it's not a big long strand of DNA. . So I'm going to use orange, blue, red, and green. I'm going to have a big orange and a big blue chromatid. Then I'm going to have a little red and a little green. Okay? These basically, this is going to come from, like, maybe this chromosome here came from dad originally in that person, and this one here came from mom originally in that person, and this one came from dad, and this one originally came from mom. Okay? And we won't need to label these each time, and we'll come back to this later. So then in the next phase, we call this prophase 1, capital I for 1, Roman numeral 1. So that then tells you something right there. Prophase 1, does that tell you there's going to be a prophase 2? Yes, you'd be correct. All right, so in prophase 1, basically the DNA doubles in the previous in interphase. And so now, because it's doubled, we now have the X's that you're familiar with in prophase. And then what comes next? Right, metaphase, but it's a little bit different this time. So this time we have the centrioles appear. But this time, instead of having all 4 chromosomes line up along the middle, what you're going to have happen is I'm only going to have, because I only have 4 chromosomes, I'm only going to have 2 spindle fibers this time. And the 2 big ones are going to be on the same spindle fiber. Remember, one's from mom, one's from dad, but they code for the same traits. And both of those are going to be on the same spindle fiber. And then the 2 little ones, one from mom and one from dad, are going to connect on the same spindle fiber. So see, they're still lined up along the middle. Okay, but it's just that the ones that code for the same traits are on the same spindle fiber. So this orange one, say that this one has the hair color gene on it. Well, the blue one does too, but the orange one came from mom and the blue one came from dad, or I can't remember which one I said earlier. Okay, but one of them came from mom and one of them came from dad. Maybe the red and the green one have the code for eye color on it, and so then. But one of them came from mom and one of them came from dad. So that's where the homologous pairs come in. Okay, so what's the next phase? Oh, whoops, we need to label this phase. Metaphase 1. Then on the next one is anaphase. All right, anaphase 1. So the whole purpose of this is just to make sure that each cell doesn't get a copy of the same pair that codes for the same traits. So we still have our centrioles, and remember now our spindle fibers have shortened because they're going to drag back the other one. So now I have the big one here, a big one here, a little one there, and a little one there. That's anaphase 1. Then what comes next? Right, telophase. But notice we still haven't split the chromatids yet. We just split the chromosomes that code for the same information. So now we're going to draw our 2 membranes are going to come back. And technically we're going to, the pinching starts happening in the middle. So I'm going to kind of add a cell membrane here And then this gets kind of crossed out. And I have the orange the big orange X up here and a little red one. And then I have the big blue one down here and the little green one. And they don't really go outside the nuclear membrane. I'm just doing that so that you can see. Alright, and this one is called telophase. So the cells are starting to get ready. Okay, and then of course, cytokinesis. Draw some membranes, some nuclear membranes. Big X, little x, a big X, and a little x. The interesting part with this is that this is random. Okay, it doesn't have to be all mom's genes in one cell and all dad's chromosomes in the other cell. It's random. That's how brothers and sisters end up with differences between each other. So this one's actually going to go by two names. This is cytokinesis 1, but it's also prophase 2. You can just write that out here. So now we get our next one. Oh, I don't have a lot of room to write at the bottom, so I'm going to write it on the side, but you can write it underneath. This is metaphase 2. And I've got my nuclear Gets that. I've got my spindle fibers and my centrioles. Oops. Let's try that. Make that a little bit better. Like that. And then I've got my big orange one is lined up along the middle. My little red one is lined up along the middle. See, they're lined up along the middle. And the big blue one here. And the little green one here. Okay, so they're lined up along the middle. What comes next? Right, anaphase 2. So I've got my centrioles, and my short little spindle fibers, and then half of the orange one goes this way, half of the orange one goes that way, half of the little red one. Okay, remember, these are now different because some of these genes came from mom and some of these genes came from dad. So we're going to end up with 4 cells that are genetically similar yet different. And then half the blue one goes this way and half the green one goes this way. I know, I'm drawing outside my cells, but it's just so that you can see. And the name of this one, of course, is anaphase 2. And what's the next one? Right, telophase 2. Alright, so now with telophase, we're going to have 2 nuclear membranes, and I'm going to kind of start pinching in the center. 2 nuclear membranes and start pinching in the center. So based on the number of nuclear membranes, how many cells are we going to end up with? Right, 4. So in this one, I have a big orange, a little red, big blue, little green, little green. And the last phase is, of course, cytokinesis, which I did not leave myself enough room to write. So I'm going to write it over on the right. You can write it on the bottom. Cytokinesis 2. And I need nuclear membranes in each of these. And I need chromatids in each of these. But notice, it's only half of the original one. So in the first one, look back at number 1, you see that there's a red, a blue, a red, orange, and a green chromosome in the first cell. They had all 4 chromatids. Now these cells only have half. It could be just random which half they get. So it could be that, the blue one gets with the red one next time, or the orange one, and you have 23 pairs of these, so the combinations are pretty much endless. All right, and then these would end up being egg or sperm cells. That's how we make them. All right, so remember, on the left-hand side now, you have a job to do. Okay, you need to write 3 questions over here that pertain to either the notes that you took here or the drawings that you did on the back. Remember, you have 3 options. Option 1 is that you would write a possible quiz question over there on the left-hand side. Option 2 would be a question that you still have about the notes. Option 3 would be a question that goes beyond the notes that you're wondering about. You can mix and match the different options. You can write all quiz questions. You can write 2 quiz questions and 1 option 2 question. Mix and match them up. And then I will be collecting this on its due date. See you in class.